I'm indeed Jens, and I'm going to present my research. I'm actually going to provide a guide to apply the medical imaging technique, positron emission tomography, as a non-destructive tool to image plants. So on the one hand, there are people here that are more familiar with the imaging part, the medical imaging part, and other people will be here that will be more expert in the plant part. And so now my presentation is going to bridge both worlds actually to study plants with this medical imaging technique. So, but first I'm going to give a short introduction about medical imaging. It's what medical imaging can do. So on the one hand side, you have structural image, where it is actually the goal to visualize a structure. So for example, you have radiography, which uses X-rays to visualize a bone fracture, for example. You also have the 3D variant, CT, or computed tomography, which uses the same technology, X-rays. And there is also a non-radioactive version, which is called ultrasounds, which uses pressure waves to visualize structures. And last but not least, you have magnetic resonance imaging, or MRI, which uses magnetism and radio waves to create a 3D image. So these techniques, all of these techniques are mainly used to visualize structures. So not exclusively, but mainly. And on the other hand, you have functional imaging. One example is positron emission tomography, which uses gamma rays to visualize actually what is going on. So you emit something into the object that you want to study, and you want to put the object in a sort of camera or detection system, which visualizes the tracer that you injected into the system to eventually follow what the tracer is doing into the objects. Another example is SPECT, single photon emission computed tomography. And today's talk will, of course, be focused around positron emission tomography. So what do we use? We use positron emitting isotopes in PET imaging. What are these isotopes? I'm going to give an example using carbon. So the core of a carbon atom has 6 positrons, shown in red, which define that it is a carbon atom, and 6 neutrons, which have no charge, and the positrons have a positive charge. So in total, there are 12 units, which describe the mass, and the 6 positrons describe that it's a carbon atom. So nature would love to play with it, and it allowed that some neutrons could be added. So if we add a neutron, it stays a carbon atom because the number of positrons stays the same, but it increases with 1 unit of mass. It's a carbon-13 atom, and nature is perfectly fine with it. But we could also subtract a neutron, and then we get also a carbon atom because it has still 6 positrons. Protons, but the mass is now reduced to 11. And this core has actually an excess of positive energy, and the core can't handle it. So what it does, it becomes radioactive, and it converts one of its protons to neutrons. So we get a different core. It's a boron atom, and it emits its positively charged as a positron. So this positron is actually emitted with kinetic energy, and it travels a certain distance, which we call the positron range. This is very important to keep this in mind. So once the positron has a reduced kinetic energy and comes across a free electron, according to the law of our good friend Albert Einstein, the mass of both particles is converted into 2 gamma rays that travel in opposite direction. So if we put this positron emitting isotope in a ring of detectors that is able to detect these gamma rays, both of them, then the system can calculate that in this line there should be a positron that met an electron, and so both are converted to gamma rays. So note again that only the position of where the positron is converted to gamma rays is detected, and not the specific position of where the positron started to exist. So if we put this into practice, I will give an example by injecting the head of a little bug called Mark with radioactivity. And so we see that his head is radioactive. So at time point is radioactive, so it is emitting gamma radiation, which can be detected by the ring of detectors, by the PET camera. So at time point 0, Mark is here. 1 minute later, he's there and still emitting radioactivity. 1 minute later there, and after 3 minutes, he crossed the plane. So in the end, we can see, or we could follow Mark on his journey towards the bed that he was moving on. But the resolution is actually not that good. So it's not clearly, or we could follow him, but it's not entirely clear that it was his head. And also notice that only his head is visible and not his body, because only his head was made radioactive. So to conclude, we could actually obtain dynamic data as we could follow Mark. PET allows in vivo experiments because Mark was very much alive, and the only drawback is that the resolution is somewhat poor. But positron emission tomography can be complemented with positron autoradiography. And in this techniques, this is actually the 2D variant of PET. We place Mark, which can perfectly done after a PET experiment. We place Mark on actually a phosphor screen. And this phosphor screens measures or detects the positrons. So here, this technique actually needs the positron range, because the positron needs to escape from the place of decay, which is in Mark. What we see after 10 minutes of positioning Mark onto the phosphor screen is that Mark is very happy and that he had actually a very good resolution. So the positive thing is that it's a very good resolution, but no dynamic data can be obtained, as it has to stay this for 10 minutes, and it's quite destructive because there needs to be very close contact of Mark against the phosphor screen. So now I have a question for the audience. If someone could answer this question. So here I have 2 images. One is from PET, and the other is taken with positron autoradiography. So can someone help me out? Which one is which? And by comparing the resolution? No? Then I will...
Please do not be shy.
Okay, I will show if I will reveal it. So the resolution of PET was worse than autoradiography. So this is the PET image and this is the autoradiography image. So why should we now use this imaging techniques to study plants? Well, because actually most of the commonly used isotopes are relevant to study plants. So you have carbon, which can be used to formulate carbon dioxide, which can be used to study photosynthesis and sugar transport. You have also nitrogen to study nitrogen uptake, and also oxygen to study water transport. And then the strange one in here is maybe fluorine, but this isotope is actually commonly built into a glucose molecule in the clinical setting. So we can also study sugar transport with it, and it can also be used to trace water, as it is as an ion dissolved in water, so as a fluoride. So another advantage of why we should use PET to study plants is that these isotopes, all of these, have a short half-life. So they actually allow repeated measurements on the same plant. And there is also no need to kill the plant or destruct the plant afterwards. So this is a very big advantage of this technique. Then if we have a look at the PET camera itself or the PET system itself, if you want to start applying this imaging techniques to plants, you can knock on the door of an imaging facility or either a laboratory animal facility, which has the laboratory animal PET, or you can knock at the door of a hospital, which has the clinical PET. So each of these have their advantages and disadvantages, and I'm not going to like say all of this, but it comes down to the laboratory animal PET has actually a small bore size, so tiny structures can be imaged. You are confined by the space that's inside the scanner, whereas the clinical PET has actually a more broader space that can be imaged, but at a resolution that is somewhat worse compared to the laboratory animal PET. And of course, the ideal situation would be that there would ever be a plant, the plant PET be constructed. And last Wednesday, I heard from the colleagues from Italy, the group of Professor D'Ascenzo, that they actually constructed the plant PET that was able to acquire PET images in the field. So it's very promising. So, if you want to start performing PET experiments on plants, you should be aware that this actually requires a lot of teamwork. So of course, it starts with a plant PET ID, which have to be communicated with all involved parties. And you have to take into account that you're working with radioactivity. So there should be, of course, some radiation protection. Then, once you selected a specific tracer, it starts with the production of the radioactivity in a cyclotron unit. Then the radioisotope is created, and a radiopharmacist then formulates this radioisotope into the desired tracer, whether it be carbon dioxide or fluorine or radioactive water. Then this radiotracer is delivered to the PET scanning room, and then where you can perform your experiment. After the experiment, image reconstruction is done, and afterwards you can process your image and quantify what is visualized. So some things to take into regard is the scan time. So the scan time is actually limited by the half-life of these isotopes. So if you want to trace carbon-11, take into account that the half-life is 20 minutes. And it's generally regarded that you can scan 2 to 3 hours with it. Other things that you have to take into account is that your tracer should be taken up by the plant and should be metabolized by the plant within your scan time. And also the dynamic range of the PET scanner is, of course, important. So as a rule of thumb, I put it here that the dynamic PET scanning, so if you want to trace the tracer in function of time, this is appropriate if the metabolic process of interest alters the tracer's distribution within 10 half-lives of time, which is already quite long and will depend on both the scanner and your metabolism, of course. Then you also have two sort of detector designs. It's planar PET detector designs, which use opposite detectors to create 2D images. This is mostly done by a group in Japan. And of course, you also can create a ring of detectors. Then you have a 3D image of your tracer distribution. Then other things to take into account is that there are image degrading effects. So in the ideal world, our tracer is decayed and 2 gamma rays are emitted that travels 180 degrees opposite to each other, which is called a true coincidence. But there are a number of image degrading effects like photon non-collinearity, scattered coincidence, and random coincidence, which all degrade your image. And actually, you need to be aware of it, but you cannot do something about it, as these effects actually determine the performance of the PET scanner. And these are measured with a phantom. In a sense, or essentially, this is actually a water-filled volume which contains tiny spheres, and these spheres are filled with the radio tracer. And then this is the image that is captured then, and this is used to calculate the performance of the scanner. So another thing which I talked about already quite some time is the positron range. So it's actually the distance that the positron travels from the decaying nucleus. So this is important. Why? Because this is in the order of millimeters. So let's say that you want to study, for example, nitrate uptake into plant leaves. Take into account that the positron range is like somewhat 2 millimeters, but that the plant leaves are by default less than 2 millimeters, so that most of the positrons will escape your leaf and will not contribute to the eventual image. So this is something to take into regard as well. It's better to use than fluorine or something. Of course, it cannot be used to study nitrogen metabolism, but it's things that you should keep in mind. So after your experiment is done, there is an image reconstruction step, and you end up with a PET image. So if you reconstruct, you did a scan for 2 hours, then you can reconstruct an image that contains all of the activity that was detected by the scanner during this period. And this is called actually a static PET image. I mainly use this image, or these images are mainly used because they everything what is in the scanner is clearly visible. Here you could differentiate the branch from the petioles on the leaf blades. And then if the desired things are visible in your scan, you can go over to perform a dynamic reconstruction, which exactly comprises parts of this complete scan and shows the tracer distribution in these time frames. So here in the first 20 minutes, you could only see the leaf blades that were labeled with carbon dioxide, and within the next 20 minutes, the tracer is being transported towards the branch. Then now we actually know what PET imaging is and what it can do. So now I'll give 2 examples so that it is more put into practice. So what was done here in this experiment is 11C carbon dioxide was used to label a wheat plant to actually study sugar transport from the leaf to the ear, so the grains. So as a first step, you should introduce carbon dioxide into the closed bag, because most facilities do not allow you to just emit a radioactive gas into the open. So it needs to be done in a bag. Once the carbon dioxide is inside the bag, there is a process called photosynthesis, which under light converts the carbon dioxide that is taken up by the plant, and it's converted to an 11C photosynthate, or a sugar. So the carbon dioxide is now as a sugar inside the plant. Normally, I label the plant for 7 minutes, which allows a tracer uptake for 80% or something. So then it's safely to remove the bag, put the plant in the PET scanner, of course, have some lights in it so that it can maintain normal functioning, and start the PET scan. So this is what the experimental setup looked up like. So we have an air inflow and outflow. So it's a semi-closed system. And this was the inlet of the carbon dioxide, with the plant here on the leaf here. So what we saw on the PET image was this. Now an image should be displaying. Yeah, I'm going to play it or try to play it again. Or what in a sense what was shown was actually the leaf was labeled, and over time we could see that there is a hotspot in the beginning here where the leaf was labeled. Then the tracer is transported inside the leaf, which is connected to the stem somewhat over here, and then the tracer is transported from the leaf stem to the ear, essentially. So what can be done is in image analysis, we draw a certain confined boxes that we call region of interest, or an ROI, around the places that we're interested in. So in this case, this was an ROI drawn onto the labeling site. And we can see as a function over time that the activity decays on this spot. That's logic because the trace or the sugar that is inside the plant is being transported towards the grains, which are actually attracting the sugar. So we can also draw an ROI onto the stem, and we see that the sugar is actually just passing by. It goes up and then goes down again. And if we draw an ROI around the ear, we see that there is, in the beginning, there is nothing, and sugars are gradually coming into the ear of the wheat plant. So most studies or PET studies, this is the endpoint of most studies. So they image a plant and they end up with time activity curves. And so this is called TAC data, showing the activity over time. But we can actually go one step further. And now this is time for a second example. And here we use actually carbon dioxide, so the same molecule, but not as a gas, but it is dissolved in water, as can be seen here. And we put, of course, a branch into this radioactively labeled water. And we also have some light that the plant can perform normal functioning. And what we see was the following. So note that only a small part of the branch is inside the PET scanner, and this is what is shown here. We see that actually most activity retains at the base, but some of it is transported. So from a physiological perspective, we can actually already know which processes are happening inside the plant. Of course, there is transport from this place to this place because we see activity. So the tracer should, in the first place, be transported. But as most activity retains here, also the tracer is captured. A part can be stored as carbon dioxide, so the molecule dissolved in water, or this molecule can be photosynthesized. It can be converted to a sugar as well. And also a part can actually be affluxed to and leave the system, so it can be affluxed to the atmosphere. So again, we can perform ROI analysis by drawing cylindrical boxes close to each other, and we can compute the activity that's what's inside these boxes, and then we end up with time activity curves. But now we go one step further. So we have the system here, and we know which processes that are taking place. So we can actually define a compartmental model describing what is happening. So you have transport. So carbon dioxide should be transported in each of these cylindrical units. A part can be captured. So with a fraction A can be stored as carbon dioxide. And from this compartment, it can either be photosynthesized, so converted to sugars, and a part can leave the system, so leave the branch. And this is part C. So actually, our model here is described by 4 parameters. So speed at which the carbon dioxide travels, and these are exchange fractions. A, B, and C, each describing one of these processes. So this model can actually be described by a set of differential equations, which are taking place in each of these ROIs. So if we have a look, what we acquired from the image data was actually discrete, so points, not connected points, of the activity inside each of these ROIs. Then we have our model, and using model calibration, a computer program tries to estimate each of these parameters, so the velocity and the exchange fractions ABC. And then to calculate eventually also the tracer activity in each ROIs, and this is given by the model fit. So if all goes well, your model fit, so the continuous lines, actually describe what was measured with the PET image. So now we have actually values from this model for each A, B, and C, and the velocity describing what is actually taking place into the branch. So from this result, it could be seen that a part of the tracer, or the biggest part of the tracer, around 50%, was transported by the water stream inside the plant. There is actually a fraction A that is, or a fraction that is assimilated to sugars, which is also the second biggest part, 40%, and a small part leaves the system, and a tiny part is stored as temporarily stored as carbon dioxide. So the main advantage of combining modeling with PET imaging is that you can actually describe physiological processes that are taking place into these plants without destructing the plant. And this is, in my opinion, modeling and PET imaging goes hand in hand, because it's very difficult to acquire this information, this physiologically relevant information, with actually other techniques without destructing the plant. So this is, a very big advantage in my part. So now I have given two examples where 11C, or labeled carbon dioxide, is used, but also other tracers can be used to study plants. And two examples are shown here. I'm not going into detail into these images, but in this study, I used 13 nitrogen labeled nitrate to study uptake in maize plants. So the roots were labeled with this tracer, and after a period of 1 hour, autoradiography was performed to see the tracer uptake into young maize plants. And you see that clearly a distribution is visible inside the plant. Note again that the resolution is quite high because this is autoradiography. Unfortunately, we also performed PET imaging, but like I said, 13N, or the positrons that are emitted by nitrogen 13, have a long positron range, and they actually, the biggest part was left the leaf, so it did not contribute to the PET image. So actually the stem was also put into PET scanner for 1 hour, but hardly anything was visible because of the positron range. And then another study that was performed by our lab is that we studied foliar water uptake, so water uptake from the leaves, to visualize uptake and redistribution of water inside the stems. And here you can see a PET CT image, PET, of course, showing the distribution of the tracer, and the CT image clearly visualizing the plant leaves. So this is also a nice example of these two techniques that can be used together to create a better picture of what is going on. So with this, I would, these are actually two examples, but there exist numerous examples which are summarized in a review paper by Schmidt et al. From 2021. I guess, which summarizes all plant PET experiments that have been performed in the past. And if you are excited and want to perform PET experiments yourself, then I wrote a guide to do this using 11C carbon dioxide or other tracers in this nice article. And if you have some questions, you can ask me all the time. Thank you for your attention.